Hey everyone, welcome to today's presentation on basic cloning workflows and troubleshooting. Um, in the previous videos that you've watched, you've talked about molecular cloning fundamentals from like a very broad point of view. In today's presentation, we're going to focus in on how to actually work with the DNA itself. We're talking about intuition, right? We're not going to talk about like how to specifically do every single step. There's no point in teaching all these different numbers for microliters to add to each other because honestly, if you do troubleshooting, you're gonna have to modify these numbers anyways. So there's no point in memorizing them. What's more important is understanding um, the reason behind why you're doing certain steps in this cloning workflow to put together DNA. And by understanding these steps, you can then be better equipped to troubleshoot when things do go wrong. So today's presentation is prepared by Andres and myself. You won't see Andres, unfortunately, but he did help me prepare some of these slides. So let's get started with the lecture outline. So when you think of cloning, you might think of the idea of making copies of a living organism. The same way for Dolly the sheep, they made like a clone of a Dolly. What we're focused on in terms of developing synthetic biology skills is more of molecular cloning. We're going to focus on bacteria and we're going to focus on E. coli. Why? Because this is a model organism. It's a good place to kind of get your feet wet and really explore how to manipulate DNA so that you can then go on to explore non-model organisms, mammalian systems, even yeast systems to do even more things with your synthetic biology skills. Now on the top right, this is a electron micrograph image of plasmid DNA to kind of get a picture of what it looks like in your head when you're working with it. Remember, we're focusing on developing intuition. Having an idea of what you're physically working with, even though you can't see it with your eyes, is very helpful. Working with plasmid DNA is nice. When you're engineering E. coli in terms of DNA manipulation, you could engineer the genome itself, but one, it's a bit more complicated. Two, it takes more time. Three, genome engineering leads to kind of these more permanent changes to kind of get your feet wet and learn about how to manipulate DNA. It's better to work with plasmids because manipulating plasmids, you can do it more easily outside of the cell itself. So it's a little simpler. Working with plasmid DNA, even though it's simpler, is actually still extremely useful, right? For example, if you wanted to produce insulin to help treat diabetes, for example, one way to do it is to get a plasmid designed to express genes, to express proteins from, say, human cloning DNA, cDNA. If you're going to put the human DNA into this bacterial plasmid to produce this recombinant plasmid, this is what we call a construct. You can then put this construct into bacteria like E. coli, and it will, one, it'll reproduce this plasmid to mass produce it, and two, it'll produce whatever is on that plasmid as long as the gene is properly structured. And from that, you can then produce insulin, purify the insulin, and bada boom, you got yourself a biotherapeutic, right? Obviously more complicated than that, but what we've done in CSBerg is simplify this kind of simple cloning workflow into something called the 5 C's workflow. It sounds like a workout program, but it's not. This simple cloning workflow we're going to teach will use restriction enzymes to help break apart the DNA and then put it back together in a way that you want to put it back together for expression, for example. And so the 5 C's workflow starts with getting the DNA so you can actually work with it, right? Collecting the DNA. So we'll talk about inoculation, mini preps, and PCR. And then once you have your DNA, you want to cut it so that you can then later stitch it back together. And this process of cutting the DNA, we call it digest using restriction enzymes. And sometimes these digests are not the cleanest things. You'll get byproducts. And so you have to do some gel extraction, some column prep to clean your DNA. In the fourth step, you're actually now finally cloning the actual DNA itself. So you're putting the DNA back together in a way that will allow for a certain application you want. And then to mass produce that, constructed DNA that you've made, you transform it into bacteria, into E. coli, to then replicate it for you. And finally, you want to check that you made and put together this DNA properly So you then have to send it for sequencing. So we're going to talk about all of this step by step, going over the reasonings behind certain things. There are 3 knowledge checkpoints in this presentation that you can pause the video at, because I'm going to give you some scenarios that you have to actually troubleshoot yourself on your own, and then we can go over them after you're ready. After today's presentation, you're ready to be a DNA wizard, just like Harry Potter. Okay, so for this presentation, I encourage you, especially if you're someone who appreciates the visual aspects of learning, to create a flow diagram. What you see on screen right now is an example of what a flow diagram could look like that can be segmented into the 5 C's workflow. You begin by collecting the DNA. We're going to focus on getting a block of DNA copied, so this fragment A, as well as getting vector B from like a glycerol stock, we call it. Once you collect your DNA, you can then cut the DNA, clean it up a little bit to prepare it for assembly, which then is the cloning step where you assemble it using a ligation, transform it into bacteria to mass produce that finished construct. And then you want to make sure that this finished construct has been put together properly. So you're going to check the DNA using sequencing. You could use restriction enzyme digest or colony PCR to do the check as well. But sequencing is kind of like the foolproof way of checking, because you can actually see letter by letter whether like your DNA has been put in the right sequence. And we're going to assume we're working on a 9 to 5 schedule, 8 hours a day, to kind of give you a sense of how long these different techniques take. Remember, we want to give you the intuition behind how to go through this workflow, and understanding how long it takes to do certain things is a great way to also develop that intuition. Okay, so we begin the 5 C's workflow by asking the question, where do you find the DNA? You can find it in the mail. If you order it through DNA synthesis, you could find it lying in the lab on a bench, for example, which is a sin, so please don't do that. You'll destroy the DNA. Put it back in a fridge or a freezer where you're most likely going to find your DNA. In our case, what we want to look for is the plasmid DNA right now, so that we can put our block of DNA into it that we're interested in. So plasmid DNA is typically found stored in the freezer in glycerol stocks. Glycerol stocks are quite literally stocks of cells that are resuspended in glycerol to help stabilize it in the low temperatures in the freezer. And you can grow cultures from these glycerol tubes that will have your stocks inside of them. So the first step is optional. If you wanted to get one cell carrying your plasmid, you could streak purify from the glycerol stock onto an LB agar plate with your antibiotic. This does take an extra night. What you could do is just straight up, get your sterile grow media with your antibiotic, and you can either get that one colony from the streak purified plate, or you can just collect cells straight from the tube using a pipette tip that's sterile, or a sterile wooden dowel stick that you can scrape the icicle from, and then just put that into the sterile media, grow that overnight for 37 degrees, and that should give you the next morning a dense cell culture that will have your plasmid DNA inside the cells, because there's antibiotic that's selecting for that. To get the plasmid DNA out of the cell, you have to go through a process called mini prepping. It's termed mini prepping the plasmid, we say. It takes about 20 to 40 minutes depending on how many samples you have. And there are different mini prep kits you can buy from commercial suppliers to do this. They all go through the same kind of like series of steps. The volumes might change, the buffers might be different slightly, but the steps are pretty much the same. The first step is to pellet your cells so that you can collect the cells and separate it from the grow media that you discard. The next step is 3 parts, 3 buffers. You first resuspend the cells in a resuspension buffer. You lyse the cells in the second step using a lysis buffer, and you can see it becoming more translucent. And that's because there's lysis happening, the cells are breaking apart, so light can go through more easily. The lysis buffer will contain SDS and sodium hydroxide. SDS will help break apart the membrane of the cell. Sodium hydroxide helps in that process too, But what sodium hydroxide will also do is help precipitate some of that junk material in the cell, like the proteins that you don't need because you're purifying the plasmids. Afterwards, you then neutralize the solution, and this neutralization will help separate that plasmid DNA away from that cell junk, that cell debris that you can kind of already start seeing here and here. If you centrifuge this mixture here, you then get this solution where the cellular debris is at the bottom and this clear see-through liquid is at the top. And this is where you're going to find your plasmid DNA separated already from your cell. Now, you can't just use this material right off the bat. You have to actually still separate the DNA from this solution. So what you're going to do is take this supernatant, you're going to put it into something called a spin column. And a spin column will have this, there's two parts of a spin column. There's one collection tube, which you can see this big part here. And inside the collection tube, there's your actual spin column tube. And that's where your supernatant will go inside. And then when you put this through a centrifuge, it'll push the supernatant carrying your plasmid through this filter into this bottom here. The DNA will stick to this filter. And the other remaining garbage that you don't want in this layer will be washed right through, and you can discard that part. You can then also use ethanol wash steps to clean up whatever is stuck to the DNA in the filter. The ethanol will help keep the DNA on the filter, but it'll also wash away some of the salts and other compounds that you don't need in your downstream processing of this plasmid DNA in our entire workflow. Once you've done all that, you can take this tube that's carrying this filter and put it into a separate tube that you then can elute your DNA into it. And that eluted DNA, that plasmid DNA, this is the purified plasmid DNA that you can now work with your cutting and pasting and everything. So we just talked about getting your plasmid DNA by inoculating from glycerol stock and then mini prepping the plasmid out of the cultured cell. That is the plasmid DNA that we're going to put our DNA sequence of interest into, so that that plasmid can then express that gene off of. So to get this piece of DNA that we're interested in, there's a number of ways to do it, and we're going to use a process called PCR, which takes about 2 to 3 hours to do, depending on how long this PCR product you want to create is. And PCR, there's a number of ways to do it based off which polymerase you buy from commercial suppliers, but the components to do a PCR, like the ingredients, are all the same. There is the water, buffer, primers, nucleotides, purified DNA, and polymerase. And the way they kind of come together is as such. The water and buffer are together so that you can have operating conditions for the polymerase. So these buffers will include some salts, like magnesium chloride, ATP, to power the polymerase. That allows the polymerase to work. The primers are there because they will stick onto the purified DNA that you put in there that you're trying to amplify a sequence of DNA from. So they specify where the polymerase should be amplifying from. And to do this amplification, you need to have some nucleotide base pairs just floating in solution that can be incorporated into this DNA chain by the polymerase. So the ratio to add these things depends on the manufacturing protocol for the polymerase, but generally those are the components. And the steps to go through to program the thermocycler, you can go to this website to find the more specific conditions for how to do the thermocycling. But on the top right, we have an image that kind of shows the process for each of these cycles that are kind of repeatedly done over time using the thermocycler. And each cycle has 3 steps. The first step is to denature the protein, the DNA sequence. And by denaturing it, you separate the 2 strands of the DNA. If you lower the temperature back down to anywhere between 55 to 65, depending on what the primer annealing temperature is, that will allow the primers to anneal to this separated DNA. And anneal, in this case, meaning it sticks to the DNA at its complementary site that you've designed it to bind to. If you bring the temperature at step 3 back up to 72 degrees, or whatever operating temperature the polymerases work at, depending on the manufacturing protocol, at that temperature, the polymerase will be able to stick onto this double-stranded component comprised of the primer and the original sequence, and then elongate that DNA chain based off of the complementary DNA like on that strand. And if you repeat this over and over again, using these amplified PCR products, you can then continue multiple cycles to keep producing the same block of DNA over and over again. And that's the amplification. And that's why there's 25 to 35 cycles that you do to get to, from like 1 nanogram to like 250 nanograms of DNA, for example. Now, once you've purified, okay, the nice thing with PCR is that these primers that you're designing to stick to the DNA, you can actually add overhangs to them. These overhangs can be a DNA sequence that basically codes for a restriction enzyme site that can be cut by a restriction enzyme. So you can imagine if you're going to create these blocks of DNA, you can design primers that will add restriction enzyme sites to both ends of the DNA to allow for you to cut it. And if you cut your plasmid DNA where you're going to put this block into with the same restriction enzyme sites, then you can imagine you can stitch this block of DNA into that plasmid. And that's the whole point of what we're going to talk about. Now with PCRing, sometimes you get some messy amplifications. Like you'll get your band of interest, the right length of DNA, but you might get like other longer or shorter bands just because of non-specific amplification. So you'll want to do a bit of column prepping to clean up your sample, your PCR product. And column preps take to 10-20 minutes. They're basically the latter half of the mini preps that we just talked about in the previous slides. And the way column preps work is you take your PCR product, you combine it with a binding buffer, and you put this mixture into the spin columns you saw before for mini prep. You put it in and you centrifuge it, and this mixture will pass through that white paper disc, which will adhere to, in this case, the PCR product. And you go through the washing steps with ethanol to wash out the salts, to wash out these other things from the PCR reaction, like the polymerase, like the buffer as well, which you don't want those salts in. And it'll wash it, and you can then elute it into a separate microfuge tube. And then you have your purified PCR product at that point. And at that point, you're ready to move forward with the next step, which is going to be the second C, which is cutting the DNA. Before we cut the DNA though, you need to know how much DNA there actually is in your samples before you cut. Because there is a ratio you need to add between enzymes and your DNA that would allow for optimal cutting in a certain time frame. So to measure how much DNA there is in your samples, your purified PCR DNA and your purified PCR product, you need to go through a process called nanodropping, which is very fast. It takes like 5 minutes to do even 2-3 samples. Nanodropping, the way it works, you can see the machine here on the bottom left. There is a clamp that closes down that you can see up here, and it's closing down onto this very tiny, hard to see metal pedestal here. It's like 2 millimeters in diameter. That's where this pedestal is where you put your DNA samples onto. And when you close this clamp onto the sample, gently, it photophotometrically reads the wavelengths going through the absorbance. So with that being said, the first thing you do with this nanodrop machine is to blank the machine like you would do for any spectrophotometric reading on a microplate reader, for example. You blank it, and the way you blank it is you use the elution buffer without the DNA from the commercial kit. You measure what the background noise for that one is, and you blank the machine on that. Because the next step is to then add your DNA, take a chem wipe and wipe off the previous elution buffer that you just put there, add your DNA, and then you close the clamp and read it again. And at that point, it'll give you an actual reading for how much DNA there is there approximately. And the data you're getting from nanodrops comes back immediately, and it looks a little bit like what we have here on the right side. You'll get a curve that has a little peak, and that peak is typically centered around 260 if you have DNA in your sample. And you'll see that there's two other features to this curve, the salt and the protein. These are centered around the values 280 and 230. So if you take the ratio of 260 to 280 or 260 to 230, which it gives you here, it kind of gives you an indicator of the purity of your sample. So 260 to 280 refers to the protein purity, and the ratio should be 1.8 for DNA. If it's less, it'll indicate that there is more protein in your sample than there should be, which might affect downstream processes, such as restriction enzyme digest. The other value to be aware of is the 260-230 value, which is an indicator of how much salt there is in your solution that shouldn't be there. You're looking for a value of 2. If you see less than that, it indicates to you that there might be some salt contamination from your previous stuff, like from your mini prep and the PCR column prep. So it might affect your downstream applications as well, such as the cutting. So be careful of these numbers that you should be seeing is 1.8 and 2. For the concentration, which is going to be in nanograms per microliter, the nanodrop is able to detect accurately between 10 to 2,000 nanograms, which is a huge range actually. For mini preps, for plasmids, you should be seeing about 100 to 200 nanograms. If you're like Andres, for example, one of the other instructors he got apparently 2,000 nanograms per microliter, you can get a big range depending on how much cell you're purifying from. Previously, when we were talking about the mini prep in a few slides ago, getting 100 to 200 nanograms would refer to 5 milliliters of culture. If you're purifying from mini prepping from 20 mils, you might get a larger number here, which is to be expected. For PCR products, we're typically expecting around 50 to 100 nanograms per microliter. But yeah, those are the general numbers for these nanodrops. Okay, so we just finished talking about the first C in the 5 C's workflow, which is collecting DNA. You can either collect your DNA from inoculating from glycerol stalks, and then that cell culture you get, you mini prep the plasmid DNA out of that. In our case, this plasmid DNA is what we want to put our DNA of interest into it so that you can get some expression. The PCRing of the DNA allows you to add restriction enzyme sites that you can cut with to put it into your purified plasmid vector, we call it. Now, at this checkpoint, we're going to look at some scenarios where people are going through some issues with their cloning related to this first C. So what I want you to do is to pause the video and read through these scenarios so that you can kind of help troubleshoot Brandon, Amy, and Austin we have here. So hopefully you've gone through these scenarios. And let's talk about each one and start with Braden's. So where's the plasmid? In Braden's case, he's using an expired antibiotic, which is not recommended because if you have expired antibiotics, they apply a weaker selection pressure on the E. coli. So when you're inoculating from glycerol stalk, putting into a cell a sterile LB media with your antibiotic, that antibiotic is there so that it forces the cell to keep replicating the plasmid, because the plasmid will contain the antibiotic resistance gene. If you have a weak selection pressure, the E. coli will see that plasmid as a burden to it because there's no epicillin, there's not enough there to be potently toxic. So it can degrade that plasmid, eject it from the cell, and you lose your plasmid, which would explain why he only sees 4 nanograms per microliter when he should be seeing about 100 to 200. So the lesson here is to use not expired antibiotic for selecting. In Amy's scenario, why is the protein in here? She's not using enough wash buffer to clean up her mini prep. She's using 30% of the volume she needs to be using. And as a result, she's not washing her plasmid DNA well enough, so some of that protein might still be stuck onto the spin column filter. So when she's eluting later on and doing the nanodrop, this 260-280 value is indicative of protein contamination, which is to be expected because the washing was done improperly. So the lesson here is wash properly, use the right volumes recommended by the manufacturer. Otherwise, just as a value of 260-280 at 0.24, it's pretty bad in terms of purity, but it still might not affect cutting, for example, but it might affect stuff like sequencing, which we'll talk about later on. So it's important to wash properly. In the third scenario with Austin, did the PCR not work? In his rush, he forgot to add binding buffer. Plasmid DNA sticks to the filter pretty well. It's a large molecule. Linear pieces of DNA have some more trouble sticking to the filter because they're linear, so they can kind of slide through these little crevices more easily. You add binding buffer to help change the charge of the DNA so it sticks to that spin column better. And forgetting to do so will cause your DNA to actually your PCR product, that DNA, to be sifted through the filter in your first centrifugation. So you just lose your DNA, which is terrible. So always, in this case, remember to include your binding buffer, because if you don't, you lose your DNA and you're going to get lower concentration values for your nanodrop. We're now going to move on to the next two C's, which is going to be cutting DNA and cleaning DNA. So to cut DNA, what we're going to use are restriction enzymes. And this is called a digest, kind of because you're breaking apart the plasmid, but because it's restriction enzymes, you're breaking apart the plasmid in a very specific site. So we have a PCR product that's linear, and we also have a plasmid product from our inoculation and mini prep. So we've added restriction enzyme cut sites to our PCR product, and in our plasmid, it'll naturally have different cut sites. Many plasmids that are sold by commercial suppliers will have specially designed areas in the plasmid that you can cut with very specific enzymes to put stuff into. So in our case, that you can see on the top right here, this is an example of cutting the linear piece, the PCR product on the top, the blue piece, and then putting it into the plasmid, the gray piece. And you can see the specific base pairs actually lining up. Feel free to pause the video to take a look at it, but that's how a restriction enzyme digest allows you to stitch together different parts as long as you cut it correctly with the right enzymes. Now, to do this cutting, the protocol will depend on where you buy this restriction enzyme, but generally the recipes are the same and the procedure is the same. So the first step is to just combine your ingredients. It's going to be a water buffer, restriction enzymes and DNA that you're working with, so the PCR product or the plasmid. Again, the water buffer mixture is there to help provide the right conditions for the restriction enzyme to operate, and it's going to operate on the DNA. This digest will take about 3 hours at 37 degrees. It really depends on how much DNA you're cutting. So for example, a good benchmark would be if you're cutting 1,000 nanograms, 1 microliter would be good if you're going to cut for 3 hours. You can play with this ratio according to the nature of your own DNA, but that's a good place to start. The results that you should expect, at least for the plasmid, for example, plasmids will range from anywhere between 3 kilobase pairs to 12 kilobase pairs. In this case, in this gel that you see down here, let me just turn on my laser. In this, what you see here, this is like a simulated analysis of the size of these DNA pieces. These are called, I call them at least diagnostic gels, or diagnostic agarose gels, and they allow you to kind of run your DNA to check for the size of the DNA piece. So what you'll always see is on one lane, we call them, is a bunch of bands that look like this, and they have predetermined sizes. It's kind of like your ruler that you're using to check your DNA samples, which would be on the right side lanes onward. Now, after you do the digest, for 3 hours, you can skip right to the next step, which is cloning, like the 4th C with ligation. But what I would recommend, at least when you're doing it for the first time ever, is to do a diagnostic agarose gel to check that you've done your cutting properly. And it's really important to do this because you might get very messy results, as you'll see in a sec. So to run the agarose gel, the steps are pretty straightforward. You prepare the agarose gel, which is just a mixture of agarose, which is this polymer, with water and some buffer. Once you it's kind of like making Jello actually. The same way you add hot water to this powdery mixture and then it jellifies over time. It's the same thing with agarose gels. And you'll kind of cast this gel into this little tray, and this tray you can put a comb, we call it, and these comb you can put into the gel itself, and it will form these little wells. I'll show you a picture in a sec. And these wells are where you put your DNA sample into, you turn on the electrophoresis machine, and then you just let it run for anywhere between 30 minutes to an hour. And that you can analyze under UV, which would altogether take you 2 hours to do everything, from preparing the gel to actually running the gel and looking at it under UV. So the results that you might get look a little bit like this. As I said before, you get the ladder on the far left lane, and then on the right side lanes, you'll get your DNA samples. Now, say this is the ladder, and say this one down here, this is the intended fragment size, a banding size that you wanted to look for, but you can see that there's other stuff on top of it as well, and this could be unspecific or non-specific amplification or digesting. Sorry, not amplification, it's a non-specific digesting of your plasmid DNA. And the same thing can happen as well for PCR results. PCRs, if you get it perfect, you should be able to see perhaps this lane here. There's not a lot of other bands, but unspecific amplifications result in this kind of mess. So at this point, you have a decision to make. Do you just column prep your digested DNA, or do you do something that allows you to specifically obtain this band and get rid of this stuff? So this will depend on your judgment, because these non-specific, unspecific banding patterns might affect downstream applications like ligation, and it might not, because it's at a very low concentration. So it's up to your judgment. My judgment in this case would be to do what we call a gel extraction, just because you do see very distinct bands that indicate that there's other DNA segments in your sample that could mess things up. Same thing with the PCR product. It'd be worth doing a column prep here at least. So this then moves to the next C, which is going to be cleaning. So to clean the DNA after doing your digest, you kind of have two if statements. You can either, if it's specific amplification, if it's specific digesting, you can just go straight into a column prep. You don't have to extract. We're going to say this target DNA banding size. If you do get what we saw in the previous gels, which is unspecific digesting or amplification, this is where a gel extraction could be useful, and these are kind of the timings for each of them. Now, column prep, it's not too different at all from what we've seen before. Again, you just add your DNA with binding buffer. This could be the PCR product that's been digested, or the plasmid DNA product that's been digested. Combine it with binding buffer so that you can put it through the paper disc in the spin column, and then wash away again the junk with your ethanol wash. And then finally elute your DNA into a separate microfuge tube that you can then work with and nanodrop to get the concentration. So this is the same as before. The gel extraction is kind of the new part. And the first part of the gel extraction is the same as the steps for actually running your diagnostic gel. It's to prepare the agarose as you would before. Prepare it with the wells so that you can add your dyed DNA samples into the wells, and then you run the electrophoresis as you normally would. The difference now is after you finish the electrophoresis run, so after your DNA has gone through, instead of using UV to analyze it, there's a special thing called like a blue light machine, we just call it in the lab. Basically, it shines a special blue light wavelength that illuminates the DNA that you would cut out. And you can see in the images up here, you can literally see the bands that you're looking for, and you can see a little bit of smearing, which might represent like unspecific amplification or digesting. So literally physically with a razor, you can cut out like little blocks from each of these lanes, and these blocks will be kind of like removing your target DNA from all this smearing and non-specific bands. And when you cut it out, the next step is quite similar to a column prep at this point. You take that chunk of DNA, and what you do is you mix that DNA with a special type of binding buffer. There are kits for all these things, and these kits will include a special kind of binding buffer that lets you help you melt the chunks into a liquid form. And then you can pass that through your spin column, collect your DNA, after washing, quite similar to a column prep. Okay, so we just talked about 2 of the C's in the 5 C's workflow, cutting and cleaning. To cut the DNA, we're going to use restriction enzymes to cut the PCR product ends and to cut the plasmid so that you can stitch that PCR product into the plasmid using the complementary sites that you've created by actually cutting it. Now you have to clean it in some cases if you get some very unspecific of cutting, and so you would use a diagnostic gel to check for that. And if it's very clean, just go for a column prep. If it's dirty, which often happens, you do a gel extraction. So here we have 2 different scenarios with Victoria and Pavan. I encourage you to pause the video now and read through these scenarios and help them troubleshoot their issues. Okay, so hopefully you've paused. Let's go over Victoria's situation first. Why is it completely smeared? And what you can see in this scenario is an image of her gel results, her diagnostic gel result. So the thing to look at is how much of the enzyme she's adding given a certain amount of DNA. So she adds 1 microliter initially to 800 nanograms of DNA. This is pretty standard. But she leaves her bench to go help someone, comes back, and then forgets how much she added. So she adds like 5 microliters, like a saturated amount, because she forgets how much like DNA there was and how much she added. And the smearing you see is a result of what we call star activity. So enzymes are sometimes able to cut sites that are not exactly where they're supposed to cut, like they're slightly different in the base pairs, which is natural. The way to prevent that is to not use too much enzyme and to not cut for too long. In this case, she didn't cut for too long, it doesn't specify, but she did add like too much enzyme, which can result in overcutting of your DNA. And depending on the activity of this enzyme, it can go really badly. So in this case, the EcoR is also expired, as you can see here. And because it's expired, it's not a fresh enzyme. There might be issues with it from storage over time. And so using that much expired EcoRI is probably the reason why. So lesson learned here is to, again, just follow the manufacturing protocol and try to use non-expired stuff. With these enzymes, even after their expiry date, they often do still work. But at that point, if it's past the expiry date and you see something wrong, the expiry date should be part of your troubleshooting, in which case you just buy a new enzyme. So in the second scenario, is my DNA clean or not with Pavan? And so in this case, Pavan is gel extracting her PCR products, despite the fact that it might be like a very specific amplification. This is commonly done just to make sure that you're not getting any any contaminated DNA at all. That's the wrong band size. And so she notices that in this nanodrop result that her 260, 230 value is particularly low. What does that mean? Well, 260, 230 refers to the salt that is in the DNA sample. So low value in this case indicates that there's some salt contamination. Now the question is, should this be expected, right? Should you expect there to be more salts than there normally is? And the thing with gel extractions is that you're using high temperatures, using a binding buffer to melt these blocks of DNA that you've cut from the gel itself. And the agarose that you're using to do your electrophoresis, it contains salts. So is it expected that there's more salt contamination than there normally would be for a plasmid DNA nanodrop or like from mini prep? I would say yes. You should be expecting more salts because it's hard to kind of get rid of everything with the amount of salt that's used in your agarose gel. So you should be expecting like not perfect numbers for your gel extraction products nanodrop. And the concentration you should be expecting is actually going to be not 50, not 200 nanograms per microliter, but more like 11 to 20 at most for a gel extraction. This is to be expected. Okay, let's move on to the next C in the 5Cs workflow, which is cloning. All right, in the 4th C, we're cloning the DNA. Now to do any cloning, the DNA has to be put back together. It has to be assembled properly, right? So because you've cut it before and you've cleaned it up a little bit using the diagnostic gel / the gel extraction. To put it together, we're going to use ligation, and then once you've put it together, theoretically you can just use that like assembled product, but it's at a very low concentration and you want to get more of it. So you have to go through a process called transformation to kind of clone that plasmid DNA into E. coli and then grow your E. coli on this plate that you see so that there's many of them. And these different colonies you can then check individually to make sure that, or rather check individually to see which one will have your properly assembled plasmid. So let's dive into it. Okay, so to clone the DNA, you have to put it back together first, because previously in the cut and clean steps of this 5Cs workflow, you've cut your PCR product, you've cut your plasmid to have both of them have overhangs. These overhangs will kind of stick back to each other. So what you can see on the right side here is an example of, let me turn on my laser pointer again. An example of this blue piece, this is, you could think of as the PCR product, going into your plasmid that you've mini prepped and cut. And they kind of can come back together just through the overhangs complementation. So in this case, AATT will be complementary with TTAA. So through hydrogen bonding, they'll kind of stick back together. Some people like to just take this kind of product, like they'll combine their PCR product with their plasmid, and then just transform that, which we will explain in a second. I would recommend not doing that because it's very likely your plasmid can just kind of fall apart. And it'll fall apart because it's only bound to each other. The target gene and the plasmid, they're only bound to each other using hydrogen bonding. The sticky ends will complement, we say, that you can see in the bottom left here. They'll complement with each other, but they will not be covalently bound to each other. The 5' to the 3' end of the G, the nucleotide here, is not covalently bound to the 5' phosphate group of the A here. What you need to do is add ligase to covalently link together the hydroxyl and the phosphate groups on these nucleotides. Only then is it covalently bound and stably put together, and that at that point you've pasted your DNA properly. So the protocol to do this is, again, like many things we're talking about, it depends on the manufacturing protocol, but typically a ligation will be consisting of a water and buffer put together that provides the conditions for the ligase to operate. In this case, it's going to need ATP in that buffer. And this ligase will then operate on the cut DNA that you add in. And this cut DNA includes your PCR product and your plasmid that's been cut. The time it takes to do this ranges. It takes about 15 minutes to 16 hours. The reason why there's such a large range is because some ligases are advertised to, be quick ligases, they call it, and process the DNA very quickly. If you trust that, it takes 15 minutes. I, for some reason, don't trust that, so I do the old school way, which is just leave it overnight or for 16 hours and let it ligate overnight at room temperature. And this takes, more time, but it's never failed on me. Now, the important thing is the ratio of the cut DNA you should put in. And this ratio is referring to the vector to the insert, the plasmid to the PCR product. So 1 to 2 is a good ratio for when you have really large PCR products that you're trying to put into the plasmid, really large target genes that you're trying to put into the plasmid. So 1 to 2 makes more sense. So you have like twice as much insert as the vector. But in other cases, the target gene, the PCR product, might be very small compared to the plasmid you're trying to put it in. And you want to increase the likelihood that these pieces will be able to go into your plasmid. And that's why you add more of it in a 1 to 10 ratio. So to determine the ratio, there are some online tools, which I'll show you in a sec, that will allow you to determine what ratio to put them in. But at the end of the day, you're putting together your cut PCR product and your cut plasmid, and you're just ligating it together so that you can transform it next. Now, again, you're following a manufacturing protocol to do all this stuff, and typically you will be incubating at 16 to 25 degrees if you're going to do it overnight. And when I say 25 degrees, I'm talking about like, I just put together this reaction in a little PCR 200 microliter tube, and I just toss it in my drawer, and let it sit overnight, and it's never failed me in terms of properly ligating the product. But yeah, it might be incubating at 37 degrees if you're doing the 50-minute protocol, just to speed up the reaction for the quick ligase version. So I just talked about restriction enzyme cloning, right, for the 5Cs workflow. The timing it takes to do this is as follows. If you've been recording it, it takes about 1 day to prepare the DNA by collecting it, because you have to inoculate overnight and then mini prep it. The cutting can take place in 1 day because it takes a few hours. But if you're going to ligate it overnight, which is what I do, it takes 1 extra day. Once you've ligated it, the next thing you would do is to do a transformation, which I'm going to talk about in a second. This process takes 1 day to grow overnight, and then the next day, the next morning, you then have to continue doing more stuff to check to make sure the DNA has been properly assembled. This whole process takes 4 days. Now, a faster way to do this it is called Gibson assembly, which I've introduced in previous videos. And Gibson assembly basically takes your cutting and your ligation and puts it into one single step to make it faster. That you can see here. So you still need to collect your DNA, but to put it together, you kind of have a single one-pot reaction, they call it, to cut and ligate back together so that you can then transform. This takes off an extra day, which kind of saves you a lot of time. And I want to go into what Gibson assembly is in more detail, which you can see on the left side here. So Gibson assembly, it's another way to put together DNA, right? And so let's just think about 2 pieces of DNA we need to stitch together, part A and part B here. Part A and part B are designed using primers to have these overhangs. The same way restriction enzymes can be added onto your PCR product. You can also PCR products that have Gibson ends. And in this case, the Gibson ends are just sequences of DNA that, in this case, you're adding to the end of part A and the beginning of part B. When you put these 2 fragments together, these 2 parts together in a Gibson assembly master mix, we call it, it contains all the enzymes and buffers needed in a single volume. A reaction will go as such. So you start off with your 2 parts kind of lining up with each other. And there's an exonuclease that will kind of chew up the DNA and kind of break it apart in the direction of the 5' to 3'. So it'll cut in this direction. It'll cut off the blue part here. And on the part B fragment, the other fragment, it'll cut the red part off. And you can see by exposing these 2 different parts, you are able to kind of click together the red and the blue regions here, which are produced from the primers. And when they stitch back together, there's going to be a polymerase in the master mix that will extend from the 3' to onward to until it reaches the 5' part that wasn't chewed up. And it kind of fills in basically what the exonuclease chewed up. And just like ligation, it has to put back the DNA together. And so there's a ligase there that kind of stitches everything back together. And bada boom, at that point you have your part A and part B stitched together using this complementary region. And then that's it. And you compare that to the restriction enzyme cloning, where you have to do the cutting and the ligation separately. The Gibson assembly method, or the Gibson cloning method, does it all for you in a single reaction. And you can think, if you have multiple fragments you want to put together, it might be simpler to just use a Gibson assembly method, because you could have part A and part B that both need to go into this linear vector, and you can add them all together. You don't need to just process A and B separately. You just do it all together at once to assemble your DNA. Whereas for the restriction enzyme cloning, you will have to cut the fragments separately and then put them together. It takes more time and effort. So the difference between restriction enzyme cloning and Gibson cloning for when to use it is when you have many parts to put together, do Gibson. If you have a few parts, like 1 or 2, like a vector and the PCR product that we've been talking about, restriction enzyme cloning is more appropriate. Either way, you will end up with a closed plasmid from ligation for restriction enzyme cloning and Gibson cloning. And that product, that closed plasmid, can then go on to transformation, which we'll talk about now. So transformations take about 2 to 3 hours to do, and then you have to incubate it overnight. This is what ON stands for, overnight, for about 16 hours. So the first step to do a transformation is to anticipate what you're going to need in the future for the transformation, which is to have agar plates. So you need to prepare agar plates with the appropriate antibiotic to select for your fully assembled plasmid. And there's two ways to do transformations. One is called the calcium chloride competent E. coli cell method, and the other one is called the electroporation method, which I'm going to compare with you right now. So if you're going to do the calcium chloride competent method, the regular transformation, I think of it as, you incubate the E. coli with the closed plasmid. So you put them together and you just let it sit on ice for 30 minutes. And this waiting period on ice will allow the plasmid to kind of like adhere to the surface of E. coli. It kind of like positions it so that it's ready for the next step. And the next step is to transfer this mixture of E. coli and your closed plasmid, your ligated plasmid, onto 42 degrees heating block. And you let it sit for up to a minute. And what this heat shock will do, we call it heat shocking because you're switching from ice to 42 degrees, it'll kind of create pores on the surface of the membrane of E. coli. And that's these holes that are created, plasmids kind of slip right through inside the cell. And you bring it back on ice afterwards to allow the pores to kind of close up again. And hopefully at this point, some cells have uptaken your constructed plasmid. And then you add grow media, some LB media, so that it has the nutrients to kind of like start growing again. Now, we compare this to electroporation, which is almost virtually the same, except the equipment is different. So previously for the transformation, you're using calcium chloride competent cells. There's a separate protocol for this to prepare these. You can also just buy these cells as well. It's expensive. There's also cells that are called electrocompetent E. coli cells. They can be the same cells, they're just prepared differently. And so for electrocompetent cells, you also combine that with your plasmid that you've ligated together. But instead of putting it on ice and then transferring it to a heat block, you're going to directly put it into what we call a cuvette, which you can see here. This is the cuvette that you're going to put your cells and the plasmid into it. So there's a little slot right here, and you use a pipette to take your mixture and put it right in between. And it'll basically touch two metal plates. And that metal plate, if you put this whole thing into this little region here, it'll apply an electric shock very fast. You just press the button, shocks, that's it. And this momentary shock will induce pores on the surface of the membrane, which will also allow your plasmid to kind of slip right into it, into the cell. And then you put it back on ice very quickly after you've added grow media, some LB media. So step 4 is kind of the opposite. The point is just to kind of close the pores back together and add the nutrients it needs to kind of start growing a little bit more. Very importantly, you're adding the grow media after you've electrically shocked your cells for electroporation. The reason for this is because if you have salts, like a lot of salts, inside your E. coli closed plasmid mixture, these salts will allow the electricity to flow through you can think of it as more efficiently. So efficient that it'll actually create an arc, like an electric arc, through the cells, which will kill the cells. You want to make sure that there's no grow media or anything like that, any salts in this mixture before you electrically shock it, because you'll kill your cells. So that's why you add the grow media afterwards instead of before. This is important for troubleshooting. Now, in both cases, after you've iced it and added grow media, the steps that follow are exactly the same. You take these cells that have this additional grow media inside of it, and you incubate that for 37 degrees for 1 hour. The reason why you do this is because the cells have just uptaken your plasmid, but they're not ready to be introduced to antibiotics right away because they're not expressing those antibiotic resistance genes that would allow it to survive in ampicillin, for example, or kanamycin or something. So you give it this time, this 1 hour time to grow, replicate that plasmid, and express the antibiotic resistance genes from that plasmid to prepare for the next step. And the next step is to take that small volume of cell culture, basically. You pellet the cells. It'll be a very small pellet, and then you resuspend that pellet in a smaller grow volume. So typically what I do is I'll grow like 1 milliliter of this mixture of closed plasmid cells and grow media. I'll grow it for 1 hour, and I'll just like pellet that. And when I dump and discard that grow media, I don't discard all of the grow media. I just do it very quickly, and I'll have like a little volume left in the tube of grow media with your cells. I just resuspended that and then transfer that solution onto an agar plate that you can then spread with whichever method you want. I use like little glass beads to do the spreading. You can also use like a hockey stick to spread. Everything is sterile, of course, to spread it whatever way works for you. And once you have that agar plate with your cells on it that have been heat shocked or electrically shocked, you then put that in the incubator and let it grow for overnight at 37 degrees. And the next morning, what you should see are colonies that grow in the plate. And each colony theoretically represents one cell that had uptaken your plasmid, hopefully. Now, the next morning when you look at what we call your transformants, when you're looking for your transformants, these are the individual colonies that grow on the agar plate that theoretically should have your plasmid that you've constructed and assembled. You're going to ask the question, what counts as the transformation worked or not? Hopefully you see results that look like this. There are numerous individual colonies that you can easily check in the next step for the 5Cs workflow. You might get something that looks more like this, more sparse. And oftentimes, if you have a very challenging like digest, which typically happens when you have large genes that you want to put into a plasmid, and you create this massive 12 kilobase pair plasmid, those are the hard ones. The bigger things you're trying to put into E. coli, the harder it is to do a transformation, as you can physically imagine, right? If the holes that you create through the electroporation or the calcium chloride method, the holes you create aren't unlimited in size. They're only going to be, there's a physical constraint for how big these holes get. If you have a massive plasmid you're going to put inside of it, the likelihood is much lower for it to go inside the hole successfully, right? So you'll get less transformants for more challenging transformations, and sometimes you'll get like what appears to be no transformants, but if you look very closely, you might actually see something that looks like a successful colony. Theoretically, you only need your assembled construct to go into one cell for this entire thing to work, for this entire copying, cutting, and pasting, this entire 5Cs workflow. It's kind of funny. And when I look at this, for example, I would think, it's good enough. If I inoculate this one little colony into some sterile LB, grow it overnight, and there's a dense culture the next morning, okay, then that would allow me to do the final step in the 5Cs workflow, which is to check whether there's actual properly constructed plasmid in that one colony or not. So that brings us to the next final step in the 5Cs workflow, which is to check to make sure that your pasting of your PCR product into the plasmid worked the exact way you wanted it to, and that it ligated and transformed properly. Now we're going to use sequencing for this because it's the most foolproof way to kind of check for your assembly, because you're reading the DNA sequence of your plasmid with your PCR product inside of it. Your insert is inside it. So let's go into it. Now, to check your DNA, you're not using any particularly new technique. Everything that you're doing now is going to be the same as before. So you're going to have the next morning a plate with hopefully colonies on it. These are your transformants. What you're going to do is inoculate these transformants, each individual colony, into LB grow media. So if you have 10 colonies on your plate, I would recommend doing 10 colonies. If you have a lot of transformants, in the hundreds, simply just pick 10 that you want to, pick 10 equally around the plate that you want to analyze, because it doesn't make sense to analyze all 100. You only need one to work, so just try 10 first. So you grow those, and you grow those transformants in your media overnight for 37 degrees for 16 hours. The next morning, will hopefully have your cell cultures and their dents, indicating that they're able to grow in the presence of this antibiotic, meaning that that plasmid that you're trying to put into the cells successfully made it in. You're going to mini prep this the same way you did like many slides back. You're going to mini prep these cultures and get the purified plasmid. This purified plasmid now is the one that contains your insert, hopefully. And what you're going to do is elute this plasmid DNA that you've constructed into a separate tube, and you're going to send that DNA sample to a sequencing company to do the sequencing for you. And the sequencing results look a little bit like this, that you can see on the right side here. On the top, we have SnapGene, by the way. You're going to learn how to use SnapGene in the course. This is SnapGene. On the top, you see your DNA that you've, this is your DNA file, right? It's like a Word document. This is your DNA file that you have on hand on SnapGene. And what SnapGene can do is take your sequencing results. The sequencing companies will send you back an email with your results. It can take your results and compare it to your expected result. This is kind of like your theoretical versus your observed, right? And you can see here that there's a mutation. It's supposed to be, I think, a C, but it's actually an A, which indicates that through your entire workflow, and at some point in this workflow, there might have been an error that led to a mutation. And you can see for the other parts that there's no mutations. So this is how you use sequencing to check to make sure that your plasmid has been assembled correctly the way you want it to. Sometimes these mutations, or at least apparent mutations, are there not because there was an issue in your workflow, but they're there because your original sequence that you put into SnapGene, your original file, it might have an error instead. So it's worth investigating whether you have the correct DNA sequence in your SnapGene program, or if it's actually because of some issue in your workflow that you can detect using sequencing. The little peaks that you see in the bottom here, this is what we call the chromatogram. So the sequencing results you get back will typically include a chromatogram in addition to the sequence itself. And these chromatograms can be useful because each peak, and these different colors, you can see there's 4 colors representing A, T, C, G. Each peak represents the likelihood of that particular color, that particular base pair, in being in that position. So sometimes it's useful to look at the chromatogram because you might see in a sequencing result that it's supposed to be an A, but instead the sequence results give you back N. N represents any nucleotide. So the sequencer at the company, the machine itself is not sure whether it's one of 2 base pairs or one of 3 base pairs or 4. The chromatogram can tell you, can give you an indicator of which ones are there. So in this case, for example, you see there's a green peak here for A. If you were to get an N instead of an A over here, you can look at the chromatogram and see which peaks and which colors are present. In some cases, you might see both a green and a black peak, for example, overlaid on top of each other, which tells you that your plasmid, maybe there's 2 versions of it in your sample. One that has one that has A for green, and one that has black, which is for G, right? That might happen, and so you can use your chromatogram to interpret your results. Okay, so to do a sequencing reaction, it's going to use a primer. Not in the sense of a PCR reaction, but these primers will allow the machine itself, the sequencing machine, to know where to start the read, to know where to start producing your sequencing information. So let's talk about primer selection really briefly. Say we have here a sequence of DNA, and this is representing an area in the plasmid that I want to put my PCR product into. These 2 black arrows represent the 2 restriction enzymes that I'm going to use to cut my plasmid DNA to put my insert into. So it's going to be BGLI, so I call it bagel 2, and PAC-I, or PAC-1. So I'm going to put my insert between these 2 restriction enzymes. So I'm going to cut with these 2 restriction enzymes. Now, say I've done the cut And I've gone through the whole workflow to paste my PCR product, in this case called MTA, with a tag called PDT3. This yellow-orange sequence I've put into the site now. You can see bagel still on one end and pacI still on the other end, right? If you want to sequence this result and check to make sure that you have this inside your plasmid after transforming, the primers you need to design cannot be right here. It can't be right in front of the MTA or right behind the stop codon. The thing with sequencing is that you need primers that are a little further out, because the first 100 base pairs from sequencing results are typically like really messy. That's just the nature of it. So you want to have primers that are further out, perhaps down here in the white region, such that when the sequencing reaction happens, it'll kind of go through this it goes through the sequence here that you don't really care about, and then it'll properly read this point onward to actually make sure you have your insert inside. Same for the reverse. You want to go outside of 100 base pairs from where you actually want to sequence to make sure that you don't like read this part here and this part here as junk, which would happen if you had a primer right here. Okay, great. So we just went through the last part of the 5Cs workflow, which is cloning and checking your stuff. So let's go through this knowledge checkpoint number 3 and pause the video so you can read Christian and Andres' scenario. Okay, so hopefully you've paused the video and looked over the scenarios. So let's go through them one by one. With Christian's scenario, false positives, he's using something called the NE-bio calculator. Such a weird name, NEB biocalculator, which is one of the tools you can use to determine the ratio of insert to vector that I mentioned before. So in this tool, it will prompt you to put the numbers for the size of the insert in the vector, the PCR product and the plasmid that's been cut. And it'll give you back the ratio to use. Now, what he should be getting is a 1:10 ratio, but what he's put in instead will give him a 10:1 ratio because he's kind of reversed them. And this 10:1 ratio means that he's going to have 10 times more vector than insert. Usually for these small 200 base pair inserts, you want to have way more of them. So you want a 1:10. What happens ultimately is that when he does the ligation with a 10:1 ratio, depending on how he cuts his plasmid, he'll actually ligate the plasmid back onto itself without the insert. So he's reclosing the plasmid without getting anything inside. This is what we call like an empty vector, right? If you transform that empty vector into the cell, it can still use it to survive in the antibiotic presence. So when he does when he sequences the transformants and finds that all of them don't have his insert inside, that is kind of to be expected from this kind of error, because these vectors will close on themselves and still be used by the E. coli. So lesson learned here is to make sure that you don't mix up your vector insert information when you're working with these tools, because it'll mess up the overall efficiency. The second scenario with Andres, did I just kill all my cells? So in this scenario, he's using electroporation because he has a very large plasmid, 12,000 base pairs. This is what I would do too. It's more highly efficient, right, to get the large plasmids inside using electroporation. Now, the issue here is very subtle. And what it has to do with is when he adds the LB to replenish the nutrients for the cells to grow. He adds it before electroshocking. And the salts that you can find in LB, there's a lot of salts. There's probably like 500 millimolar sodium chloride. It's a lot. And it's going to arc the cells in the cuvette itself between the metal plates. And this arcing is going to kill the cells. So yes, he did kill the cells. And the lesson learned here is to make sure that you're adding LB after you've done the heat shock or the electroshock to replenish the nutrients. Congratulations, you've made it to the end of this presentation. What I want to do is summarize some of the stuff we've talked about, or rather all the stuff we've talked about, again using the 5Cs workflow. So the first C is to collect your DNA, and there's two ways to do it. The first way to collect your DNA is by mini prepping it from your glycerol stock. Glycerol stocks are again where there is cells that are stored in glycerol that will already have plasmids inside of them that you can grow from. And then when you grow them up overnight, you can then mini prep the plasmid back out. these plasmids will naturally have restriction enzyme sites to cut inside, so there's no need to add it. Compared to PCR amplification. So if you want to amplify and copy a particular piece of DNA that you want to put into this plasmid, you can use your primers and design them to add restriction enzyme sites to these ends. And then you can cut both the PCR product and the vector such that they can be stitched back together in the second step for the 5Cs workflow, which is cutting. Now, sometimes when you cut this DNA, when you digest it, it'll be very messy and unspecific. So you have to go through a cleaning process sometimes, particularly a gel extraction, to get these single bands that you want to put together. And to put together, this is where we get to the fourth step in the 5Cs workflow, which is cloning. So to assemble your DNA back together, you've cut your parts, you want to ligate it back together, we say. You use a process called ligation with ligase, and then that stitched together assembled product can then be transformed into E. coli. It can be put back into E. coli so that E. coli can then replicate and mass produce that plasmid for you to work with and work from. So in this process, you can use calcium chloride chemical competent cells. You can also use electroporation to do this. Either way, you will get transformants the next day. And these transformants are individual colonies that theoretically should have your assembled construct that you can then send off for sequencing after mini prepping from one night's culture. So when you sequence these results, hopefully you will be able to get a DNA sequence that matches what you expect it to look like, which is the insert inside your plasmid DNA. And you can use SnapGene for this to kind of compare between the two. And so that's it. This summarizes the 5Cs workflow. If you have questions, feel free to comment in the section below or contact the CSBurger team with your questions. Otherwise, we hope that you can use this information in the future to kind of inform how you design some of your workflows for cloning. And also it can serve as a basis for you to use other techniques like Gibson assembly and other like cloning assembly methods in your lab space. Okay, great. Thank you everyone and enjoy your days. Cheers.